as Diplocasteria. So we have to focus on only four orders, that is Dorylampida, Triplonchida, then Aplanchida and Tylanchida. So under Diplogasteria subclass, three orders are there: Diplogasteria, Aplanchida, and Tylanchida. So what are the differences? Just to see in one slide, that in Diplogasteria, Fosoria is a type of teeth, very small teeth are present. So these are called Fosoria. And in Aplanchida and Tylanchida, stylets are present. And this stylet is very much important for the feeding of nematodes on plant roots. And Aplanchida, you know, some Aplanchids are plant parasites and some are fungal feeders. So in fungal feeders also, some are having stylets and some are not having stylet. But in Tylanchida, all of them are having stylet. The other difference is that males have no bursa in Diplocasteria. They are having 7 pairs of genital papillae. Here, uh, 3 pairs of genital papillae, and here either one pair or no genital papillae. Okay. Now, what is the difference between Aplanchida and Tylanchida? Since we will go for studying these two orders only, which are very advanced nematodes. So most of the nematodes which are coming into economic important nematodes, like for our crops, and in Aplanchida, mushroom nematodes are there because they are fungal feeders. So that mushroom cultivation is also growing in large amount in large area. So these are also very much important. And some Aplanchids are ectoparasites of the shoot part, like Aplanchoids, Fregeri, Aplanchoids, Rhismebosi, then Aplanca savini. So these are very important nematodes. So what are the difference between Aplanchida and Tylanchida? So I could not have shown you the figures, but just you imagine that both are having uh, tripartite esophagus. Tripartite means procarpus, metacarpus, and basal bulb. So these are having aplanchid esophagus and these are having tylanchid esophagus. So what are the tylanchid and aplanchid esophagus? Aplanchid esophagus means the median bulb is very strong. Here the median bulb is very strong. It may be round or it may be rectangular, and the valve is very, that valvular plates means they are having very strong muscular plates for sucking the sap, and here the median valve is not so much strong. So this is the difference between aplanchid and tylanchid esophagus. Both are tripartite. Now the second difference is that opening of dorsal esophageal gland is above the median bulb. Means all the glands are present in the basal bulb, but where they open, this is the difference in different nematodes. So this median and this esophageal gland, this DOG, dorsal esophageal gland, opens just above the median bulb. But here the dorsal esophageal gland opens at the base of the stylet. Okay. Basal knobs, or third difference is basal knobs of stylet are either lacking or poor. Shaft and knobs form metadactyons or metadactyons. So this is the origin. Just 1 minute. So sometimes like Aphelenchus ebeni, that comes under order Aphelenchida, they are having, don't have basal knobs. Only the uh, conus and shaft are there. But in case of Tylanchida, stylet knobs are very well developed, and origin of shaft and knob is different. Okay, so that is not very important for you people, because these are especially for the nematologists who are studying nematology. So you just know that the basal knobs of stylet are either lacking like in Aphelenchus aviarii, but are present in affluent wild species. And here in many nematodes will come, we'll learn one by one. Like many uh, super families are there, many subfamilies, families, then in different genera. These stylet knobs may be of different shape also. So that are all of taxonomical importance. Some are tulip shaped, some are anchor shaped, some are round shaped. So according to that, on the basis of that shape and structure, you can identify that this nematode is of this genre. So it is well developed. Here anus is like overhanging leaves, means you can see from the upper side of the upper side of the cuticle that anus is overhanging, and here it is just a pore like. Bursa is present with bursal rays. So bursa is having some bursal rays which are of sensory origin. Here bursa is present only, but bursal rays are not. Spicules are either cylinder or rose thorn shaped. Here also, this is just rose thorn shape is the peculiar character of aphelenchoids. So if you see any nematodes which are having rose thorn shaped spicules, then you should assess that this is aphelenchoids. And the cylinder shape is of aphelenchus. Spicules are cylindrical and never rose thorn shape. So if it is rose thorn, means it is especially for the Aphelenchida group. Genital papillae present, here the genital papillae absent. Now in the order Tylenchida, four suborders are there. Tylenchina, Oplolamina, Trichonematina, and Hexatylen. So one by one I will take. So first is Tylenchina. So under Tylenchina, that is suborder, this is infraorders. I have not written here. You can note it down that under Tylenchida, two suborders are there. Infraorders, two infraorders, that is Tylencata and Anguinata. So Tylencata and Anguinata, on the basis of these characters, they are differentiated. Like here, cardia is present. Cardia, you know that the wall between, the door between esophagus and intestine. So in Tylencata, cardia is present, but in Anguinata, cardia is absent. Are you getting what I am telling? Hello? Hello?
Yes, ma'am.
Are you
Yes,
getting what?
ma'am. Yes, ma'am.
Okay. If you don't understand, just tell me. Here the sperms are small, here sperms are large. Spermatheca is offset or side attachment. That is it is having less cytoplasm. Spermatheca, you just have seen that in female reproductive system there is a swelling. There is a swelling part of just below the uh, this ovary, that is called as spermatheca, where the sperms are stored. So when the egg passes through that spermatheca, it is fertilized with the sperms. So it is not in continuous in that in Tallinkata, it is just offset, means it is just like an attachment to the main reproductive system. But here it is continuous and it is having fair amount of protoplasmids are present. The tail is conoid. Here the tail is long and filiform, means Tylencata or the Tylencata have very long and filiform. Anguinata, you must have known that Anguinata, it is seed called nematode. So that comes under this infraorder Anguinata. So many nematodes are there, but they all have some amount of sexual dimorphism, means the females look different from the male. So wherever there is endoparasitic behavior, the female shape is changed. Since Anguilla tetanus first feeds ectoparasitically, then it comes inside the ear head of a wheat, and then it feeds like endoparasite. So the female shape is changed. So female swells a bit, not that much like in melatonin. So adenal bursa. Adenal bursa means the bursa is present only near the anus portion. So that is called adenal bursa and also called paloderan bursa. That I have told you in morphology class that bursa may be paloderan, uh, then arecaderon, and uh, another one is there that if bursa is present only near the anus, it is leptoderan. If it is present from the anus to the tail, it is paloderan. And if it is present from both sides, like an ear, then it is arecaderan. So here it is adenal means leptoderan. Here it is complete bursa when tail is short. Bursa terminal or subterminal when tail is long, means it depends on the tail shape. Now we will start with the Thailand kata. This is very important. So under Thailand kida, this Thailand kata is also very important. Under Thailand kata, superfamily comes Thailand quietia. So we will study the Thailand quietia. So under this superfamily Thailand quietia, four families are there. Four families are Italian kitty, Italian kitty, Tylodoridae, and Epiphydophoridae. Now we'll start with Tylodoridae, the third one. Tylodoridae. Actually, this Thailand kitty is very much important that we will learn later. So first the other ones, like Tylodoridae. Tylodoridae have only two nematodes which are important. Since this family may have many nematodes, but those nematodes which are infecting our crops are Tylodorus and Cephalanthus. See all the characteristics of Thailand kata except having very long stylet. Means all the characteristics you have seen under Thailand kata group, they all are having, they have all these characters and very long stylet. They have like Tylodorus and Cephalanthus. What is the major difference between these two? That Tylodorus have more than 75 micrometers stylet and have a label disc, and it has more than 30 micrometers stylet and no label disc. Means Cephalanthus have stylet lesser than Tylodorus. Now the fourth one, that is Epiphydophoridae. Epiphydophoridae, they are very thin nematodes. Means they are thin and long nematodes, and the genus is Epiphydophora. So whenever you write the scientific name, you have to write in italics. Okay. Otherwise, I will not give you any marks on that. So take care of writing this scientific name like genus or species name. If the name of the species is there, then it will be in italics. If you write SP or SPP, SP means single species you are denoting. If more than one species is there, you can write SPP, but that is not in italics. But if name is there like Melitoga incognita, then both the names are in italics. If you write Melitoga species, then SPP is not in italics. Okay, now the third one is A-Tylenchity. A-Tylenchity. Okay, this is third one, A-Tylenchity. We have seen Tylodoridae, Epiphydophoridae, and now A-Tylenchity. In A-Tylenchity, A-Tylenchus and U-Tylenchus. So these three families, they don't have so much nematode that are very much important. So in A Talencuti, cephalic CT is present on head. A is less than 30. What is A? So A is the ratio of body length and maximum body width. Okay. So if the body length is more than the body width, so it is very thin, then A is more than 60. So generally for nematode measurement we use different denotations that was given by D-mens, so also called D-mens value, like body length, body width, the body width at the anus portion, then stylet length, stylet width, many denotations are there. So that are of very much important for the nematode measurement. So here the two genera are present, Tylenchus and Tylenchus. So only the major difference is that adrenal bursa is absent here and here adrenal bursa is there. Okay, and A is less than 30 in our Tylenchus. Less than 13 is these nematodes are wider, not very longer. Now the first one is Tylenchus. So Tylenchus has subfamilies, four subfamilies: Tylenchini, Duosalcini, Boliodorini, and Thardini. So here the amphids are labial and pore-like, means in both these cases, Tylenchini and Duosalcini, amphids are labial and pore-like. Here amphids are post-labial and slit-like, and here it is labial and pore-like. Labial means on the lips, post-labial means behind the lips, and slit means it is a long opening. Here lateral lines 3 to 4, occasionally 6. Generally, I told you that lateral lines, that is the longitudinal lines present in the lateral sides of nematode body, is of taxonomical importance. So on the basis of lateral lines, you can identify that this nematode may be of this genera or this family. So in Tylenchini, Tylenchus and Coslanchus is there. Duosalcini, Duosalcinus and Melenchus is there. In Boliodorini, Boliodorus and Basilia, and in Thardini, Tharda. Okay, so these four subfamily have these genera. Now comes the Anguinata. So Anguinata will study only one superfamily, Anguinoidea, family Anguinidae, and genera. These are many genera. Okay, so on the basis of gall forming structures, we could classify into three. Like some fungal feeders are there, that is Ditelenchus, Mycilophagus. Some are aerial plant feeders but not gall forming, they are Ditelenchus angustus. Ditelenchus angustus, you know, this is a cause organism of paddy. Uh, this is in paddy, sometimes the earlets don't work in the infections very early. So this is the causal organism. So both the Ditelenchus is there, but they are having different species, so they have different feeding habits. Like Mycilophagus, because of the name, you can understand that Mycilophagus means feeding on the mycelium of fungus. It is fungal feeder and feed on the mushroom. So this is a very much destroying organism of mushroom. Now the gall formation, that you know that anguilla is commonly also considered subanguina, paranguina, heteranguina, mesoanguina, narcoanguina, cyniparanguina, orina, afrina, halenchus. So these all genus, these all genera are uh, they can form galls on plant parts, like some form on seeds, some form on leaves, some form on the branches. So they, according to their feeding habits, according to their different structures, they are in different genera. So what are the characteristics of anguinity? They are parasites of above ground plants or plant. They have a microbiotic state that you have studied in survival. that if the nematodes are on the above ground parts of the plant, they have to face the extreme environmental conditions like sometimes hot weather, sometimes extreme cold weather, sometimes drought is there, sometimes windy condition is there. So they have to survive through these conditions. So they undergo an hydrobiotics. So in hydrobiosis is there. They form their own house like galls on stems, leaves, flowers, and transform them into galls, and they live inside. They can keep their eggs or juveniles inside the galls to survive. Preferred plants of Graminy and compositive family. So they are the parasites of Graminy and compositive family like wheat, and then Applequards family, uh, that this flowering plants like uh, chrysanthemum, and then Applequards Lismarosae, strawberry. These are the hosts. The infective states may be J2, J3, J4, or adult. Okay. So generally the nematodes are having only the J2 stage, which are infective stage, but in some cases there are exceptions like here. The infective stages may be J2, J3, J4, or better. High fecundity to survive in extreme environmental conditions. Suppose you have taken one seed box of wheat, it may contain 20,000 to 30,000 juveniles, so they have very high fecundity, high egg laying capacity because to go for survival. Both male and female infect the plant parts and become endoparasites. I told you again. No change in style and isoflavors become either new tiling kit or the basal bulb becomes a stem-like extension. Cristoformalia have multiple rows of cells and have more than 18 cells in each row to accommodate more members. Cristoformalia, I told you, in female reproductive system, this is 4 by 4 or 3 by 4. Sometimes here you can say 18 cells are there. They are just to arrange the cells. If more number of eggs are there, then they have to be large to accommodate them. So here, because they are laying more number of eggs, so they are having more number of cells in cluster formation. Then in they are having different genera, so different genera have different characters, like in Heteroinguina, the infected stage is J3, in Mesoinguina, infected stage is J4. So this slide is not very important for you people. You just know about the characteristics of endometrium, how they form balls and how they undergo in hydrobiotic stage, what are their hosts. Okay. Sorry, one call has come. So, so this is especially that which form on flowers, which form on roots. So this is not very important for you people. Now the suborder Hoplolemina. Okay, so under this you have seen in Tylenchida is there, Hoplolemina is there, Trichonematina and Hexatylenina. So you have finished Tylenchida, now coming to Hoplolemina. So in Hoplolemina, the nematodes have very strong stylet very strong stylet knobs. So these are very peculiar character of Hoplolemina. So in Hoplolemina, they are parasites of roots. They may be ectoparasites, some are endoparasites, or some are semi-endoparasites. Semi-endoparasites means their half of the body remains inside and half, two-third part of the body remains outside, one-third inside. Like citrus nematodes, reniform nematodes, they are semi-endoparasites. Both sexes are vermiform or females may be solen. So females may be solen in which case where the nematodes undergo endoparasite or semi-endoparasites. But in ectoparasites, both male and female are having the same cell. Anterior ends may be degenerated because they have to water the roots. Well-developed head sclerotization, strong stylet, well-developed cardia, DOG opens at stylet base, vulva in the middle of the body, monoprodelphic ovary with PUS. What is PUS? It is post-uterine sac. Post-uterine sac means since the basic structure of ovary is one interior and one posterior. But during the development, during the evolution, the posterior part of that ovary is reduced to become a sac-like structure. Okay? So that's why it is called post-uterine sacs. In some parts, in some nematodes it is present, in some nematodes it is not present. So this is also of taxonomic importance. Okay? Clusterformaria, they may be 4 by 4 or 3 by 4 or irregular. Female tail cylindrical, rounded or spiked, occasionally filiform. So in Hoplolemina, the most important nematode is Hoplolemus that are generally found whenever you wash the soil. So you may get in 50 to 60% of that soil samples these nematodes. You can get these juveniles, males and females all. And the females and juveniles are having cylindrical shaped tail. But when they become male, after development, the males are having very clear cut this bursa and it is spiked. Pachmates may be pore-like or enlarged to form scrutella. This is also very important. But one important characteristic that stylet knobs is very strong in Hoplolemina, and other is that the pachmates are very large. It is called scrutella. Male tails are convoid. That I have told you that male tails are not round. It is convoid. Bursa is complete. Then under Hoplolemina, two superfamilies are there, Dolichodoroidea and Hoplolemoidia. So in Dolichodoroidea, males have equally developed stylet and esophagus, and here males may have more developed stylet and esophagus. Okay, so you just remember what are the Hoplolemina's characters. Okay, what are the characters of Hoplolemina? I will ask about this 4 to 5 questions. Like you are having only practical that I will ask you one that theory part will be from external. So I will give you all the slides. You just go through it. So if they will ask some different questions, you may be able to answer. Then superfamily Hoplolemoidea, and this is Dolichodoroidea. So you just remember this is a family of Hoplolemoidea, Hoplolemiti, Platylenchidi, and Heterodactyly. So Hoplolemina is very important suborder in which you may find the most important nematodes like in Hoplolemiti, Hoplolemus, in Platylenchidi, Platylencus and Rhodalencus is there. In Heterodactyly, what are the nematodes? That are most important nematodes, that is Melidothine. Cyst nematodes, they are present in Heterodactyly. So these three nematodes have this slide is very important. You just go through this. Hoplolemiti, they may have ecto or migratory ecto, or and few forms are semi-endoparasites. But in Praetalionchidi, all are migratory endoparasites. In Heterodactyly, maybe semi-endoparasites or
Ma'am, slide
endoparasites.
is not changing.
It's not changing.
No, ma'am.
Hoplolemity, pretalinkity, heterodarity. This slide is where you are able to see.
Yes, ma'am, not visible.
These three columns you have seen. So in Hoplolemidae, normally both are vermiform. Here all stages are infective. In petalincity, just remember that if the nematodes are endoparasite and migratory, means they are moving inside the roots from here to there. So they are having all the stages are infective stages. So at any stage they can come out of the roots, and at any stages they can come inside the root. Okay. But in sedentary endoparasites or ectoparasites or semi-endoparasites, only the second stage of the nematode is infective. But in some cases there are some exceptions, like in citrus nematode, fourth stage, that is just immature females are infective stages, like in Rotylempulus reniformis. Here also you can see heterodarity J2 is infective stage, but J3, J4 and adult females are solen. That is Meladog and Heterodera, they are having sexual dimorphism, means females are differentiate. Here J2, J3 and J4 remain vermiform. Here also they are remain vermiform because they are migratory, so they have to be vermiform, otherwise they can't move. Males develop without metamorphosis. Metamorphosis, what is the metamorphosis? Means when they develop, like in case of melanodyne, both males and females develop within a case. Means they both become swollen-like structure, but the males are always vermiform inside that shape, and the females are becoming solid. But after the maturity, the males come out in a vermiform shape, and females become like they are before, swollen shape. So this is called development through metamorphosis. So males develop through metamorphosis, means they are in juvenile stages they have different shape, and in adult stages they have different shape. So here it is not through metamorphosis. Here they are not going through metamorphosis, but here males and females both develop through metamorphosis. But the males are swollen during the juvenile stages, but when they become adult, they become vermiform. Now the hoplolemity, this is not important for you. Then subfamilies of hoplolemity. So here this slide is also very much important. In Hoplolemini, what are the genera? Hoplolemus, Scutellonema, and Aerolemus. So Scutellonema, that I have told you that phasmids become very large and called Scutella. In microscope, you can easily identify that Scutella. So this nematode, Scutellonema, is having very large phasmids. So the name is also Scutellonema. Although Hoplolemus is having large phasmid, but that is smaller than the Scutellonema. Aerolemus, this is a predatory nematode, very thin and very fast moving nematode. Rotylenkini, Rotylenchus and Helicotylenchus. Rotylenchus and Helicotylenchus, they are also very much important. Helicotylenchus, the name itself suggests that they are spiral nematode. They are uh, the common name is spiral nematode, and Rotylenchus is reniform nematode. So why the reniform nematode? Because their females look like kidney shape, so they are called reniform nematode. And in Helicotylenchus, all the nematodes, whether it is juvenile, male or female, all are in a spiral shape like 6 in 6 digit. After death also they are taking that shape again. So this is called Helicotylenchus. Aphasentylenchini. This is Aphasentylenchus and Rotylenchulini, and Senegalodema. So all these nematodes come under this Hoplolemidae family. So they are all having different characters. We should not go through this one. Now the Praetylenchidae. The next family is Praetylenchidae. This is also very much important because Praetylenchus, that is lesson nematode, Hirschmaniella, this is root rot nematode of paddy. Only this nematode is able to survive in flooded condition. Then
Ma'am, again the slide is not changing.
it is happening. I am not keeping in display mode because of this. Now subfamilies of Praetylenchidae.
Yes, ma'am.
Okay. So in Praetylenchidae, two subfamilies are there, Praetylenchini and Nacobini. In Praetylenchini, Praetylenchus, Hirschmaniella, and Rhinophorus. Rhinophorus is also called burrowing nematode. This is a very notorious, you can say, notorious nematode of all the plantation crops like banana, areca nut, and then black pepper, and on different hosts they produce different symptoms. Okay, in banana they are causing toppling disease, that all the whole plant topple down after infection, and after infection of this nematode the fungus, fusarium species and bacteria, they all attack on these plants. So the the pseudostem, that rhizome part of that banana, rots and it topple down. In black pepper, all the leaves become yellow, so it is called yellow disease. And in areca nut, in coconut, all the plantation crops are affected with this nematode. So this is also called plantation crop nematode. And because of their feeding habitat, because um, they are migratory, and they when they migrate, they produce burrow-like structure and move forward, so also called burrowing nematode. Petalincus is called lesion nematode because they produce lesion wherever they feed. But this is not continuous, so you can say uh, in scattered structure, lesion structures are there in plant roots. That Hirschmaniella is also called lesion type symptoms. Then in Nacovus, it is also called false root knot nematode because the female becomes swollen. So these are the different structures. If you can remember, it's good. If you don't want to remember, then only you have to remember the different uh, families. You can make a flow chart, like if different type of, like here, if the hoplolamina is there, say here under hoplolamina, hoplolemoidia. So you can keep one heading like hoplolemoidia, then make a flow chart that it is having three families, and under these families how many subfamilies are there and how many genera are there. So according to that you can remember easily. Okay, then come to dolichodoroidea. So after hoplolemoidea, another superfamily is dolichodoroidea. Dolichodoroidea nematodes are very large and thin nematodes, and they are ectoparasites. Okay, they have long stylet to eat deeply in the roots. So they are
My
ectoparasites of
slides are
roots.
not changing.
Now,
No, ma'am.
dolichodoroidea. Stylet length depends on the part of roots they feed. Esophagus is tylencoid or hoplolemate. So esophagus is called tylencoid or hoplolemate or tylenkid type. Tylenkid type esophagus means generally what you have learned that tripartite esophagus is the normal that is typical esophagus. But hoplolemate type esophagus means there is some slight changes. What is the change? That change that basal bulb is slightly hanging over the intestine because of the large size of esophageal glands. So
Ma'am,
if
we are
the
unable to see the screen. It's not visible.
should I see her again? Is it there? Dolichodoroidea?
No, ma'am.
Okay, let me share again. Now is visible?
Yes, ma'am.
Okay. So, no sexual dimorphism because they are ectoparasite, they don't need to produce more number of eggs. So, both male and female are uniform. Well developed cephalic sclerotization, stylet length depends on the part of roots they feed. Is they under this superfamily, some nematodes may have a small stylet, some may have very long stylet. So if they have to feed on the epidermis, they are small in stylet. If they have to feed uh, on the inside part, that is in different layers of tissue, then they have to be long stylet. So esophagus is talented or not that I am telling you. In talented, normal esophageal bulb is there, but in hoplolemmy, the esophageal bulb is hanging over the intestine because it is having large size of esophageal glands. Media is quadricolony. Quadricolony means 4 by 4, that is 4 row of cells and 4 column of cells. Tail is cylindrical or spiked or maybe filiform. Means tail is either cylindrical or they have some spike-like structure and they may be filiform, long and sharp. Tail is conoid and bursa is complete except in some forms. Now the families of Dolichodoidea. Slide is visible.
Yes, ma'am.
So in this superfamily, four families are there: Dolichodoides, Silenchidae, Tylotylenchidae, and Velonolemnidae. And they are all having one genera that is of importance: Dolichodorus, Silencus, Tylenchorhynchus, Velonolemnis. So whenever you pronounce in Tylenchorhynchus, some may pronounce it like ch as ch. So don't pronounce ch as ch. It is ko. Tylenchorhynchus. Velonolemmus, Silencus, Dolichodorus. So they all have different structures, different characteristics. Now the family Tylotylenchus. They have Tylotylenchus. Tylencorhynchus. Tylencorhynchus is very much important nematode. Then Dolichorhynchus, Merlinius, Trophurus, and Trichodylynchus. So they have different structures. On the basis of this, they are divided into different genus, like they have different lateral lines. The lateral lines are same, they may have different esophagus overlapping. So I told you that Helicotalancus it is very much important. So under suborder Haplolamina, family Haplolamidae, subfamily Protylencoideae. So whenever you have to write the systematic position of any nematode, like if I ask about uh, Helicotalancus, that write the systematic position of Helicotalancus, then you have to start with the phylum. Phylum Nematoda, then class Shanincia, then subclass, then order, then suborder. Okay, then superfamily, superfamily. Fertilization and females. Okay? So if you see, I told you that in melatonin, most of their production is without males. But males are found there. Sometimes women are found, but the males are very long and they have the spicules at the very end of the tail and really well developed stylet and healthy fertilization. Males develop my metamorphosis. I told you. Eggs are this means they the rectal glands in the digestive system. They release some sticky material just to hold the eggs in protective manner. Just to after whenever they get any host, they hatch from that eggs. Otherwise, they remain inside. Semi-endo or endoparasites. Females become swollen, lemon shape, pear shape, or kidney shape. Means if they become lemon shape, it is heterodera. If it is pear shaped, it is globodera. If it is swollen, it is melanocyte. If it is kidney shaped, it is different nematode. Females have annuals or lace-like pattern. Annuals or lace-like pattern. This is cuticular ornamentation. Sometimes body could become thick when female glands become cyst, like in heterodera or globodera. Didelphic ovary means two ovaries are there. Majority are diprodelphic means both the like in melanodyne both two ovaries are there and both are anteriorly directed. Some are empedelphic. Empedelphic means two ovaries are there but one is posterior. The matters may be secreted by rectal glands or vaginal glands. Eggs may be retained inside like in cyst nematode. They may form galls or may not form. Forms own food sink means when they infect the plant host, they change they modify the surrounding cells into syncytium. Syncytium means their cell membranes are broken and many nucleus come in one cell. This condition is called syncytium. Giant cells like melatonin form giant cells because one cells become very bigger. That is called giant cells. And SUGC, that is single uninuclear giant cell, this is a different nematode which are forming this type of giant cell. J2 hatches are from the esophagus haplolamid type means the esophageal bulb is hanging over the intestine. Conoid tail, Hajimid tail, core like or maybe provided with a lens-like structure. In the middle of the body, there is genital primordium. Genital primordium is just the cells which are going to produce the female organism. Okay, so this is very deeply written. It is not much important to you people. Now, these subfamilies Nematode, Heteroderinium, and Velidoderinium. This is very important slide because you are going to identify that nematodes, that Heterodera, Globodera, Punctodera, Captodera, Dolichodera. These all are cyst nematodes, and these all cyst nematodes come under subfamily Heteroderinium under the family Heteroderinium. And under the family Heteroderinium, Melitogyninine. This is the subfamily under which melatogynin species are there, and this is the nematodes. This is good, not all nematodes, and these have almost all the plants are, means more than 95% plants are their host, and they are adapting more plants. They are having Melodonema and Nicopodera, Molitonematodes, and they are having vermiform fingers. Here in Ectoderiny and Melatoderiny, fingers are swollen. I told you the cyst nematodes and root knot nematodes. They are endoparasites, means completely sedentary endoparasites, means when they come inside, they have one place to their whole, for their whole life cycle. The cuticule is straight off at every stage of molting. This is the only difference, means major difference between Ectoderiny and Melatoderiny, that I told you in morphology class that in Melatoderiny, the nematodes do not shed their cuticles during molting. So the juvenile stages are not there, which is absent here in Heteroderiny. That in cyst nematodes, all the molting stages are different. Molting, they become feeding. They become feeding stages. But here, because they don't set their cuticle, they are not able to feed. So they are able to feed only in J2 stage or adulthood stage. They form syncytium or SOGC, means heteroderiny. All the cyst nematodes form syncytium or SOGC, but in melatoderiny they form giant cells and they form root knots, means the roots become swollen. So you have to remember only that. And this is also very much important. If you see the second stage juveniles of melatoderiny and heterodera, so only two differences are there, that they have melatoderine have weak stylet, very small stylet and thin stylet is there. But in heterodera, the nematodes are having very strong stylet. This, when you see under microscope, it is shining. Is it the slide is visible to all? Here the tail is pencil point. When you sharpen any pencil, that pointed like structure is there in melatoderine. But in heterodera, it is not that much pointed, but the tail is hyaline. Hyaline means transparent. The tail looks like transparent. What is perineal pattern? Perineal pattern is the cuticle pattern of females of melatoderine species at the perineal region. So perineal region is that region where the anus is found, vulva is found. So during that vulva and anal region, the cuticular pattern becomes some different like in different species. So you have to cut the perineal pattern and see the different type of structures. And according to that, you can identify the different genera like incognita, javanica, erinaria, hapla, gramicola. They all have different type of perineal pattern. So these are the perineal pattern of different species of melatoderine, that excretory pore, male head, lateral lines, frequency of occurrence, J2 length, tail length, distribution, and their host. So only the Graminicola is having only Gramini family as host, but all these species have polyphemus, and they all have different. They can feed on many plants. This hapla is of temperate habitat, means they are are tropical and semi-tropical countries. Then under Heteroderini subfamily is there. So under this, they form cyst. So how do you know that which cyst is of which genera? So you have to cut the cyst cone. So if the cyst is there, you have to cut the posterior part, which is called cyst cone. So they are having different parts. The cyst cone have different parts like mulva piece, vulval piece, under piece, body. So if you see the diagram, then you can understand what are that part. So on the posterior part, when the because cyst nematodes are host specific, so whenever they find their host, their eggs hatch and the juveniles come out. So from which part the juveniles come out? Through the cyst cone. So one, this is fenestra. Fenestra means birth opening. So they just tear that part and come outside, and that is called fenestra. And this fenestra length and width is different according to the cyst is called bifenestrate or ambifenestrate or circumfenestrate or afenestrate. So these are also very important if you can remember that. So this is fenestra. So fenestra is the birth. Now this is different type of cyst nematodes like Heterodera, Globodera, Punctodera, Captodera, Dolichodera. Globodera is not having any cyst form because they are very much round and they are the place of potatoes. Heterodera is based on many plants, and different diseases are attacking. Like Heterodera G is attacking on wheat, Heterodera Ebony is attacking on wheat and barley. Likewise, this Punctodera, Cactodera, and Dolichodera, they are not very much important. Only the Heterodera and Globodera is important. In the Globodera, two species are there: Globodera pallida and Globodera rostropiensis, but both are parasites of potato. So here the difference between rostropiensis and pallida. See here the different species like Gabeni on wheat and barley, Phillips Zabi on wheat and barley, Morphy on Kanas, Kazani on the cube, the Texas, in G, Mage or Vetiver, Sorghai on sorghum, Graminees on Bulgras, Dinosaur on Dactygon. So they are having different types of structures. So don't go through all this. You have to remember only the half type of Heterodera or Globodera is there. This the major difference between this slide is important for you. What are the cyst cone? How the Heterodera and Globodera are differentiated? What are the hosts of their Heterodera and Globodera that are important for you? Then some non-cyst forming Heterodera, some non-cyst forming Heterodera are there like Melododera and Verrucas. Okay, so only these two are important nematodes. Then Crappodera, Rhizonema, Atalodera. These are all, but they are not very important. Okay, so in this class, I will go for Glyconematina. I will send all these slides to on the mail or on WhatsApp group also. Just go for it. The next one is Glyconematina. Actually, Glyconematina nematodes are having very highly ornamented cuticle. So because of this, these nematodes could not move very fast because they are having very large hanging type. Sometimes they are having spines, sometimes they are having flap-like structures, so they are ectoparasites mostly. Some are semi-endo or endoparasites, but they are very sluggish nematodes. Okay, so I am closing here. So any question or anything you want to clear? So next class will be of Friday. If you can give me extra classes, you can arrange, or otherwise I will take on Friday. So on Friday this class will be finished. Then will be next week, interaction and nematode. Okay, so in two classes, we try to finish it. The introduction part is completed, morphology completed, taxonomy we are going through, biology we have finished, ecology we finished, symptoms we have finished. Then classification of plant parasitic on the basis of parasitism. That I have told maybe ectoparasites, endoparasites, sedentary and migratory. So only the after learning class interactions and nematode management will be there. That I will try to finish in 2 or 3 classes. Okay.